Okay, well, it's appropriately titled Bespoke Technologies for Discovering the Unknown. And a blank screen is perhaps on theme. So we have hopefully 4 very exciting speakers for you today. Without too much further ado, we have Stephan Daetwyler, we have Keng Chu, we have hopefully Spencer LaVere-Smith joining us virtually. And finally, Caroline Mollenbusch is going to give the last talk of the session. So I'll just quickly introduce our first speaker. So our keynote speaker for the session is Stephan Daetwyler. So he's an instructor at the U of T Southwestern Medical Center in the lab of Rio Retovoica, I can never pronounce that well, where he develops technologies to study the development of cancer and regeneration. So before he came to Dallas, he obtained his BSc and MSc degree in interdisciplinary and sciences from ETH Zurich, Switzerland. And after his undergraduate studies, he joined the lab of Jan Husken, who most people know as a pioneer in light sheet microscopy at the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden for his doctoral degree. So can we get the slides up for the speaker? Oh, we do have it. All right. So without further ado, please, welcome.
Thank you very much for this very nice introduction. And I also would like to thank the organizers and all the volunteers who made this meeting possible. And on Wednesday, we learned if the food is good, the meeting is good. And I can confirm it has been a very nice meeting today, but not only because of food. So today I want to present to you our work on multiscale or self-driving multiscale microscopy. And what you see on my title slide is basically we are interested in not only looking at a single cell, but we want to study a single cell in its context and how a single cell, for example, here, if we study cancer metastasis, how a cell spreads over a whole organism, but also understand what the molecular processes are that the cell undergoes for this. But to come to my talk, I would like to first introduce to you the zebrafish as model that we use for most of our questions, then introduce the hardware and software that are the foundation of our multiscale microscope, then show you 2 applications of immune cancer cell interaction and cancer survival, and then give you an outlook where we want to go next. So to the zebrafish as a model system, why are we using such a small, randomly seeming organism? And the reason behind for this is, as you can see already on this movie, as I play, we can really look at individual cells on a whole organism. So zebrafish provide a near physiological environment with blood flow, with an immune system, with different metastatic niches, for example, the head, the heart tissue, the hematopoietic niche where new blood cells are formed. And there are many genetic lines available to reveal this tissue context already that has been established by many researchers in the field. And for us in imaging, the optical translucency is sort of the selling point for zebrafish. We can really image into zebrafish, and we can use a high throughput because we can generate hundreds of embryos per week to really address our question with a statistical power. And if we want to study cancer research, we can inject, for example, human cancer cell lines into zebrafish and then image those human cancer cells with subcellular resolution. And how can we do this experiment? We can modify and we can culture our cancer cells outside of the organism. We can perturb whatever we want to do with siRNA. We can induce tags, and then we can put it into a zebrafish and study where they end up. And we can use the multiscale microscope to study cell dissemination, cell interactions, and also the morphology of those cells and their signaling. So coming to the hardware, what is the basis of our multiscale microscope? We use light sheet imaging for various reasons. And one of the reasons is that we can really rotate our sample or our complex organism and really have 360-degree access to it and optimize the angles we look at. And light sheet, and you have seen already several talks about light sheet, so I want to keep this short, provides an inherent optical sectioning because we only illuminate the fluorophores in our plane of interest. And that also translates to fast detection because all those fluorophores can be simultaneously captured by a camera chip. And that leads to minimized phototoxicity and bleaching compared to other imaging modalities, especially for live imaging. And therefore, it's ideal for non-invasive and long-term imaging over days of imaging. And we can even have a sort of a medium throughput of stacking individual zebrafish on top of each other in such a more conventional light sheet microscope, and therefore image several zebrafish embryos over days. And what you end up with in a conventional SPIM light sheet microscope is movies like this, where we can now, for example, here in this case, look at leukemic cells in circulation. And as I play the movie, you can follow the individual cells on their path through the organism, and we can study where do they end up, what is their migration pattern, and we can detect rare events. But when I started my work with Retofiolka, I was really interested in what are actually those cells doing, what is their environment, how do they interact, what is their signaling, and that's why I came up with a plan to build a multi-scale microscope. And the basis for multi-scale microscopy is basically some of it lies in the detection. So we can very quickly flip in and out the flip mirror to change from 11x magnification to 55x magnification. But it's not only the magnification that we change, it's also the resolution by employing two modes of light sheet modality. One is this classical light sheet, the SPIM, and the other is ASLM, axially swept light sheet microscopy, that I will introduce on the next slide. The principle behind ASLM is that, there is a trade-off between how long you make your light sheet and how thin you make your light sheet. But Kevin Dean and Reto Fiolka came up with the idea, what happens if we generate a very thin light sheet, but then move it back and forth and synchronize the readout on the camera chip with where only the small or the very thin light sheet is. And this basically generates a huge improvement if we now look at the axial side from traditional SFM to ASLM. And just to give you a more intuitive understanding of how we achieve this in terms of hardware. So as I play this movie, you basically see how we can move a remote mirror back and forth, which then translates in our sample space with a pivoting of our ASLM or our thin light sheet. So what do we achieve now with this? We now have a microscope that can change the detection path and also change the modality. And we end up with a low resolution light sheet of around 1 micrometer lateral and 2.4 micrometer axial resolution, and a high resolution modality where we have 400 nanometers lateral and 650 nanometer axial resolution. And you see the improvement in bead samples. But how does this translate to zebrafish? And here you basically see now on the bottom, we can capture now the whole organism efficiently with the low resolution SPIM and zoom in into high resolution with the ASLM, and you see the improvement using ASLM compared to the low resolution modality. So I was very excited at this moment. Now I have my multi-scale microscope. Now I want to study and image in vivo samples. But as you see very quickly that if I play this movie, I face the hurdle. So if I statically set a region of interest on a developing living organism, very quickly the region of view goes out of the field of view that I statically set. And this is for many reasons. It can be technical reasons. So we start with a very small field of view, but we also have sample movement, sample drift. But it's also biological, because sometimes the cells of interest might migrate out, and the fish or the organism that we image, or the spheroid, they move and they change over time. And you might now ask, why do you not image everything that is black now? And the reason for this is it would generate a huge amount of data per time point, and it would just take too long. And we heard some estimates of this from Elizabeth Hillman and other speakers this week, that high resolution imaging of everything just doesn't practical. And we summarized also this idea in a recent review that we face a volumetric imaging barrier with a single technique. So we cannot expand beyond this barrier efficiently. And what we need to overcome is new sort of smart adaptive imaging or multi-resolution imaging techniques, or also expansion to expand this volumetric imaging barrier to be more efficient in our imaging. And basically what this requires on a microscope development side is developing software for it. And basically to present software, it's hard because it's mostly lines of code. But what the software basically does is it's constantly monitoring the low resolution information and does real-time processing of the whole organism to adjust where we want to image and to take this information. And on the back of this is basically custom Python-based hardware control software that I established that does the in parallel the acquisition and the processing to be efficient. And now coming back to my title slide, what you've just seen as a still image is now a movie. So we can now really look at the whole organism, how it grows and unfolds its tail and up, and zoom in into individual breast cancer cells in this case, and there are intricate protrusions that they actually form inside the vasculature, and how, for example, you see a cancer cells migrating along a protrusion and against the blood flow towards the hook that it had in the tissue. And with this, we can now really study these complicated interactions in tissue over hours to days. So if you're interested in any of the hardware, the software, or also the analysis code of everything I basically show, it's available on GitHub, with also some documentation that you can follow. So coming to immune cancer cell interaction as a first application of the multi-scale microscope. And we have seen also, again, beautiful talks this week, the keynote on the first day, but also this morning, about the very complicated, but also very interesting behavior of macrophages. And we were wondering, can we actually use now our zebrafish disease model and the multi-scale microscope to go after those interaction in a cancer context, which is highly complicated. And macrophages can have very different functions, from anti-tumor to pro-tumorigenic behavior. So what you see here is now basically the data or the raw data where we can now look at the whole organism, and we can zoom in, and for tissue context, the vasculature is in cyan, and we can zoom in into our green cancer cells, and the macrophages on the bottom just in gray, or on top in magenta. And we can look at these interactions over many hours, and I will just show you the quantification today on the next slide of those interactions. So here, basically, we then went on to quantify how the macrophages now in color come to the red cancer cells. And as I play the movie, we can really see how macrophages leave the part where we do not have those red dots, where we do not have cancer cells, and circle around where the cancer cells are in the low resolution. And that is in stark contrast. And here you see the red cancer cells were cleared from the circulation. So they were removed. And this is in stark contrast to a control experiment where we do not have any cancer cells. And as I play the movie, you see that in the control, the macrophages patrol as much tissue as possible. They are almost uniformly spread. And we can put numbers to this by applying the Hopkins statistics, which is an indication of how clustered cells are in an environment. And we see that if we have a xenograft, macrophages are clustered around those cancer cells, and in a control environment, they're almost randomly distributed across the zebrafish. And for the high resolution imaging, what was basically not possible with this low resolution data was using just watershed thresholding or segmentation for imaging these dynamic shapes of macrophages. So we leverage a tool developed in the Danuser lab from Felix Chow, where we can now segment more easily in 3D. And you see here some example of that segmentation on complicated environments such as vasculature, T cells, or aggregates or tissue. So applying this segmentation pipeline, we were able to segment a high resolution shapes of macrophages. And here you see the cancer cell in red, in gray, the vasculature tissue context, and individually segmented macrophages in color. And as I play the movie, you can see how those macrophages first circle around those cancer cells. They do not seem to not recognize them at first, but then first macrophages start to attach, and then several come together to efficiently, or in a sort of collaborative manner, clear those cancer cells out of the circulation. You can still see some red remaining fluorophore when in the macrophage as it digests it. And out of this movie, we can now extract single macrophages and look at their shape in a more detailed way. On the top, you see macrophages in a xenograft case, where you can imagine the immune synapse towards the cancer cells, those cups. And on the bottom, a control macrophage, where you really see how they extend their arms or protrusions to really monitor as much space as possible. And again, we can look at it in 3D to really understand their cell shape and signaling. And in this case, we also see that in a xenograft case, they have different shapes than in control, so shape follows functions. And to quantify this a bit more, this shape follows function, we defined global geometric features such as volume or surface area and abstracted this into a PCA space. So each macrophage now is a great point over the whole duration, and we see sort of how macrophage shapes at hour 0 are different than our macrophage shapes at hour 16. And if we now have those distributions, we can assign a p-value, how similar these distributions are. And if we plot this over time, we can basically get a matrix of each hour to each other hour, and we can see that if it's more blue, then the p-value is high, so meaning the shapes are very similar. And if it's sort of white or less blue, the p-value is low, so the shapes are very different. And then we can sort of observe shape transitions. For example, from hour 4 to hour 5, it's quite white, so shapes have changed in this hour quite drastically. And we can recapitulate basically what we observe in the movie, that the shape follows this function from circulating macrophage to first attachment to tight attachment to phagocytosis to dissolving of clusters. Now we were wondering, okay, I presented you now U2S osteosarcoma cells. And if you look at the deep map portal, those cells do not metastasize very well, so they have a very low score. But what happens if we now put in a very highly metastatic cancer cell line in our zebrafish model? And I wonder if you can guess if I play the next movie. So again, we have our cancer cells in green and the macrophages in white. And they already start to attach, so maybe something will happen. So they again form these immune synapse and the interactions. But what you observe is that the cancer cells seem surviving, and in the end of the movie, the macrophages even leave the cancer cells again, although they had this tight adjustment, although they are human cancer cells in zebrafish. And that now basically opens up the door to actually follow this up with more biological studies. What happens? Why are those cancer cells protected from others? Is it the ECM deposition? Is it the shape? Is it the stiffness, the rigidity of the cancer cells, the membrane rigidity? Do those cancer cells shed receptors that macrophages recognize, or is it the cell signaling? Do they actually manage to polarize the macrophages that they interact with over hours? So coming to the next application, cancer cell survival in our zebrafish model. And for this, fortunately, we already had talk about lipid transport protein just this morning. So you might already recognize basically that this barrel is there for transporting lipids. And we were looking at a protein called bridge-like lipid transfer protein member, family member 2 or BLTB2. But in the cancer context, its previous name might be more revealing because it's breast cancer overexpressed gene protein, or 1 protein. And so we were really interested, why is that protein so overexpressed in breast cancer, and what is its function, or how does it affect cancer survival? And for this, that was a collaboration with Shubhra Banjure and the William Prince Lab at UT Southwestern, where Shubhra basically discovered or described how this protein is at the interface between the ER and the plasma membrane, and it's shuttling lipids from the ER to the plasma membrane, and thereby controlling membrane rigidity for cold adaptation in yeast, for cancer cell growth, inhibiting apoptosis and promoting cell invasion. And we were wondering again, how can we now contribute with our in vivo zebrafish model for this? And here you see an application where the throughput of zebrafish is one of the selling points. And so I injected hundreds of fish, and we're looking at the survival of the cancer cells in the tail. And we see that if you have a control environment, the cancer cells or the breast cancer cells, they survive, but they actually even increase in number. While if you knock down this SHBLDB2 protein, the survival drops significantly. And it's not only the survival, because we now have the zebrafish data, we can also look where do the cells end up, where do they go. And basically on top, you see the control environment at 4 hours and 24 hours, and we see that the cells spread very nicely, while on the bottom, if we knock down BLDB2, the cells do not manage to migrate upwards. And we were wondering, can we actually now apply our multiscale microscope to understand why the cells lose their ability to migrate? And basically what we saw, if we look at an actin marker, is that their cell organization, not only the membrane rigidity, but also the membrane organization seems to be disturbed, because we do not see those nice ruffles anymore that breast cancer cells have, but they seem to be gone, and the cells seem to lose their organization and thereby also their ability to survive. So coming to a quick outlook. So we are not only able to look at cancer, but also development. So on the left side, you see a movie where we have an early stage embryo and the division that those cells undergo as the embryo forms. But I presented you most work now in zebrafish, but we're also interested in other models such as spheroids, where you can, for example, image a spheroid or the whole spheroid and zoom in into the leading edge. And for example, here, see how a breast cancer cell here divides at the leading edge and forms the actin ring, while still being able to observe the whole organism or the whole spheroid. And what we also want to do or move forward is not only observe, but also perturb. So here you see a spheroid, cancer cells injected into zebrafish, and we are able with a 2-photon laser to target one single cell, and we can change it. For example, here photoconvert, but we are not only interested in photoconversion, that is good for cell tracking, but we are also interested in optogenetics to induce molecular perturbation inside in a living organism. And with this, today I show you how we leverage the zebrafish model organism to address many questions in cancer research, but also beyond, how we can use the hardware and the software of a multiscale microscope to look at immune cancer cell interaction and cancer survival and where we want to go in nearest future. And with this, I come to the acknowledgement slide. This has only been possible by the support of Reto Fiolka and Galden Stanuser at UT Southwestern, the whole Fiolka and Cheng lab, and my main collaborators, Hani Masloum Farsibaf, Felix Zhou, Dagan Segal, and Kevin Dean, and Jill Westcott, Rolf Brecken, Shubra, and William Prince, and other collaborators at the cell biology department, like Murray Carmen and Morales. And if you're interested in more of our work, we recently published our paper where we go into more details how we can look at the whole organism to study cell dissemination, migration, rare events, and really understand what the cells are doing in this context. And with this, yeah, thank you very much for your attention.